Dr. Stefan Zimmermann from the CHUV and the Cancer Center Lausanne will talk about extending beyond PD-L1, PD-1 targeting. Please.
Thank you. So good afternoon to everyone. It's my pleasure to give you this talk about what still lies in store after PD-1 and PD-L1 targeting. So I am a medical oncologist. I work in the Immuno-Oncology service in Lausanne, where we essentially do early drug development in cancer immunotherapy. So we conduct industry-sponsored and academic trials in immunotherapy. So I'll take you on a journey to look a bit at the pipeline. What is there beyond PD-1 and PD-L1 targeting? It's incredible to think that it's been almost 13 years that since the first patients enrolled into the first in-human trial evaluating what would later become nivolumab. And in these 13 years, we have now 6 anti-PD-1, anti-PD-L1 monoclonal antibodies that are approved by the FDA or the EMA in almost a dozen cancer types and cancers with defects in their DNA repair machinery. But we are all acutely aware of the shortcomings, and that there is a phenomenon of acquired resistance, but also a primary resistance. So I will start with 3 observations. The first one is that the global pipeline in IO is vastly expanding. So the authors here conducted a global survey of quite a comprehensive survey of the global pipeline in 2017, and then did a second survey in 2018, so a year apart, and these are the trends. So looking just at the number of targets, you see that there's a 67% of the agents. There's a 67% increase in the number of agents. When you break this down according to mode of action, you see that some areas are expanding much more rapidly than the others. For example, cell therapy, so this will be for the next speakers, but we have a big increase here in T-cell targeted immunomodulators and other immunomodulators. Vaccines are growing as well, albeit a bit less, and there's quite a growth here in this smallest category here. Beyond just an increase in the number of agents, there's also diversification of agents, because there's a 50% increase in the number of drug targets, active drug targets. The second observation is that we have not yet exhausted the potential of PD-1 and PD-L1 targeting. When you take a look at what is in clinical development at the moment, targeting PD-1 or PD-L1, you see this impressive number of agents. So some of these are monoclonal antibodies that will likely differ very little from what is already on the market, but there are interesting formats like bispecific antibodies that will be directed at 2 different targets with the obvious intention of improving on efficacy, but also on safety. And then there are other things like oral PD-L1 inhibitors here. The other reason we have most certainly not exhausted the potential here is that virtually all of the agents that we'll discuss later on are ultimately developed in combination with these inhibitors. And the third observation is that the late IO pipeline is still very sparse. So if we focus just on monoclonal antibodies in a pivotal trial at the moment, or as of late 2018, so this is quite a comprehensive list of the monoclonal antibodies against cancer, late 18. You do see that here there are monoclonal antibodies against PD-1, PD-L1. When you're looking for other IO drugs, you see maybe one here against LAG-3, and that's it. The others are monoclonal antibodies that are directed at overexpressed proteins on the surface of cancer cells, or antibody-drug conjugates that do not fall under the definition of what we usually consider to be immunotherapy. So there is not a huge wave of new agents coming to the clinic immediately. Now, we are all aware that checkpoint inhibitors may not work or may initially work and then fail the patient. This has been very nicely reviewed in this review. We have gathered knowledge about the mechanisms that are underlying this primary or acquired resistance, and although they do not yet inform what we have to do in the clinic to overcome them, it certainly highlights that there is ample room in improvement. For the purpose of the presentation, I will structure the different agents or targets according to where they develop most of their activity in this cancer immunity cycle. So you're aware of this concept that in order for a cancer immune response to lead to effective cancer cell killing, there needs to be these different steps that have to be initiated and propagated, with starting with release of cancer cell antigens, their processing presentation by antigen-presenting cells, the priming and activation of T cells who then traffic and infiltrate the tumor bed, recognize the cancer cell, and kill them. There is an extraordinary diversity of different targets that I have arbitrarily here placed in these different steps, and we'll take a walk around this cycle with a few examples. Most of this, I will not have a lot of outcome data. This is mostly early phase data, so I'll not bore you with toxicity data, but there are some examples that deserve mention. In a first step, there must be a release of cancer cell antigens, and this can be obviously circumvented by the use of vaccines. We'll talk about this. Oncolytic viruses to promote the release, chemotherapy, radiotherapy, we've talked about this, will allude to CD47, so these don't eat me molecules that limit phagocytosis of cancer cells and epigenetic modifiers. Now, vaccines, you saw an increase 1/3 within a year, with a distinct shift from vaccines targeting or using tumor-associated tumor antigens that are overexpressed, or cancer testis antigens, towards tumor-specific, so neoantigen vaccines in most instances, for various reasons, because the technology allows it, and also mechanistically, this is thought as the next good step, because it will actually harness a T-cell repertoire that has not been blunted by central tolerance. Now, these vaccines vary in their formulation, so they can be nucleic acid-based, or they can be whole cell, like dendritic cell-based. The adjuvants vary, and the delivery vehicles vary. There are multiple players. We'll talk about it a bit later. A usual workflow is like this. In this instance, the peptide vaccine, starting from tumor material that is sequenced, including germline information, and then there is proprietary bioinformatics, where the epitopes are selected and the vaccine designed, manufactured, and given to the patient. Now, with an mRNA vaccine, this is absolutely similar. This is an example from BioNTech. So starting from tumor sequencing material, including germline sequencing, then neoepitope selection, RNA synthesis, formulation, and treatment of the patients. You look at the pipeline, there is an incredible variety of different platforms that are used. So, for example, here, Gridstone uses a chimpanzee adenovirus vector, and then this self-amplifying mRNA, whereas Advaxis uses this Listeria-based delivery. BioNTech and Moderna use mRNA, and this is or focused on mRNA, and this is also a field that is really massively expanding of late. This mRNA therapeutics, which can be vaccines or other therapeutics. So these mRNA platforms offer quite a few advantages over other platforms in terms of costs, of course, but also potency and safety. They harness the body's own protein-making machinery to actually transform this string of nucleotides into either cancer antigens, like neoantigens in this instance here, or immune stimulatory molecules like OX40 ligand or cytokines, or even non-coding RNA, which will trigger these innate sensors. So this is a field in full expansion. Now, we have heard from previous speaker about CD47. A few industry sponsors have pulled back from this. This is a field that has known a few setbacks, that's been most active in the field of hematological malignancies. But there's still some active research with these trying to block these don't eat me molecules. This is an example from last ASCO. This is a fusion protein with an inactive Fc domain, and here the CD47 binding domain, preventing the activity of these don't eat me molecules. And just a quick example, a combination with pembrolizumab in non-small cell lung cancer, you do see some activity, albeit very modest. So clinical data is still extremely limited and mostly limited to toxicity data. Now, these antigens have to be processed and presented by presenting cells, and in order to elicit a CD8 T cell response, this has to happen. These dendritic cells, these cross-presenting dendritic cells, have to be exposed to the proper stimuli, like these DAMPs or PAMPs, which can be injected intratumorally or even systemically. One example here is STING. So what these sensors have in common is that they strongly induce type 1 interferon, which is absolutely crucial for effective cross-presentation of the antigen. So it will lead to a different acidification rate in the endosomes and more expression of MHC class 1, and the proteasome will be different. So this will really promote this cross-presentation to prime cytotoxic CD8 T cells. And so STING is a cytosolic double-stranded DNA sensor. There are other innate sensors that are being explored in the clinic. Again, very, very scarce clinical data yet. This is an example from ASCO with the STING agonist from Aduro. You do see here a few responses that are the blue triangles. It's really hard to squeeze out a message here, except that it is tolerable, that it needs to be, seems to have to be injected on a more frequent basis. So this is the weekly data set, whereas the monthly data set looks less promising, but quite difficult to squeeze out a message here. To be continued. Now, priming and activation of T cells, something that is heavily regulated. We all know of these immune checkpoints that both inhibitory or co-stimulatory signals that ultimately will define whether there is immunity or tolerance in the absence of the correct stimuli, and all these are attractive targets in drug development. So, of course, the inhibitory checkpoints were the first to have been targeted with vast array of monoclonal antibodies, so LAG3, TIM3, etc. And lagging behind is the development of these stimulatory, of agonists for these co-stimulatory molecules, such as ICOS here in the B7-CD28 family, or the other targets in the TNF receptor superfamily. For various reasons, this has been a slower development. These agonistic antibodies usually have a bell-shaped dose-response curve, so dose escalation trials are much more challenging. The design of the molecule is more complicated to get right, because you absolutely need either an inert Fc portion to not trigger a T-cell activation in the liver, for example, with what happened with 4-1BB early molecules, or actually target tumor tissue with bispecific antibodies to have a more tumor-directed activation. Furthermore, this clustering that has to happen with these agonistic antibodies is something that is extremely dose-dependent or concentration-dependent, so very challenging clinical development here. Just an example for TIM3, there was a presentation at SITC last year. So this is the one from Tesaro, again, developed in combination with their anti-PD-1. This is the spaghetti plot in NSCLC. These are all patients that have progressed on previous checkpoint inhibitor. And you do see here on the right, with a higher dose, there seems to be some promise. Once these T cells have been successfully primed, they have to infiltrate into the tumor bed, where they'll basically meet an environment that is absolutely teeming with suppressive cells and immunosuppressive signals. It's an environment that is usually hostile, it's acidic, it's depleted in amino acid, etc. And one very active avenue of research is to try to modulate this recruitment of these MDSCs, either by blocking the ligand itself, so the chemokines such as IL-8, or blocking the receptors that will on the MDSCs, so be there neutrophils or tumor-associated macrophages. So when you take a look at all these targets on this list, you have the targets here who will prevent or modulate the recruitment of tumor-associated macrophages, or even repolarize them, or for neutrophils, the same. It has been also quite a challenging journey here for this compound. So I don't have any good clinical data to show you yet. The adenosine pathway, as well as the kynurenine pathway, is something that is very actively investigated. I'll just skip here. And there is increased activity here as well in trying to deplete Tregs, or try to modulate this ratio of T effector cells and T regulatory cells. So, for examples, there have been new anti-CTLA-4 monoclonal antibodies that are non-fucosylated to increase antibody-dependent cellular cytotoxicity, or newer generations of anti-CD25 monoclonal antibodies that are not blocking the effect of IL-2. So CD25 is the alpha subunit of the IL-2 receptor, which together with the other subunits forms the high affinity IL-2 receptor that is highly expressed on Tregs, but much less so on effector cells. And by not blocking the effect of IL-2 on other cells, you actually achieve a preferential depletion of Tregs. And then there is TGF-beta, which has proven extremely challenging, with some promise here with this fusion molecule, which is a TGF-beta trap and a PD-L1 inhibitor, which is very closely resembles avelumab. If all this fails, there is the possibility of redirecting T cells in an unspecific manner, and this has been termed synthetic immunity. And this can be done through various pharmacologic means. So Immunocore has this platform where they use these ImmTAC molecules, which are stabilized T cell receptors with a CD3 binding moiety here, which will recognize with a very high affinity at the expense of specificity, will recognize this peptide HLA complex, so on the cancer cell, so including intracellular proteins, or you can use bispecific antibodies with various formats. And bispecific platforms are extremely numerous. They are asymmetric formats here, which most closely resemble normal IgG, with the advantage of being less immunogenic, but they're more difficult, more challenging to manufacture because of mispairing and purification steps. And then there are symmetric platforms or constructs where usually you don't have this problem of mispairing. These are usually less stable or less soluble. They have a tetravalent binding, so for each target you see two binding domains, which may bring advantages. And then there are fragment-based formats where you don't have this Fc portion here. This comes at the expense of the half-life, but improves tissue penetration. So they all have their advantages and disadvantages. Most of these engage T cells via CD3, but some of them engage NK cells via CD16 and a cancer antigen, like Glypican here, MUC16, ovarian cancer, HER2, etc. They all have advantages and disadvantages. This is a popular construct here. This is single chain peptide, so these half-life extended bites. So basically it's a bite with an Fc portion. Amgen is pursuing, is really resting on this platform here. And this is also a very interesting concept, these 2+1, where you have two regions binding the antigen of choice, which will highly direct the antibody to the tumor bed, and lower affinity CD3 engaging part. And this will prevent that the drug is stiffened in the lymphoid tissue upon infusion, and that you really have distribution into the tumor tissue. So there are a few molecules here that are developed in this fashion. And then the fragment-based, so these are really lower molecular weight drugs that can engage T cells or NK cells. I will skip this because I'm running out of time. So as always, for the community working in drug development, I think the biggest challenge has come from the fact that clinical development or the development of new drugs has completely outstripped what our understanding of the biomarkers or the immune environment, and we are still at the loss of identifying which are the evasion pathways that are actually altered, are they dominant, and are they non-redundant. Then we are supposed to integrate all these tumor and immune metrics, although they are limited because they are models, they are multiplexed and multiple, and the most immediate challenge in the clinic is actually to design clinical trials that will translate to the clinic, will get health authority approval, will match patients with the treatment that is most effective and are adaptive enough. And these conditions sometimes appear to be mutually exclusive. And with this, I thank you for your attention.
Thank you very much for this very nice overview. We are a bit over time, but we have time for one question. I guess that was somewhat disencouraging. Nevertheless.
Which of the 7 mechanisms do you think is the most promising? You were very fond of vaccines, but you know what? Where is the next breakthrough coming in from Lausanne?
Oh, well, cell therapies? Yes. I think it's definitely. I think it's definitely where we will see progress in these stages where all else has failed. But I think the breakthrough will probably come from the understanding of what is actually missing in the microenvironment. We know that it's heterogeneous, evolving over time. It will be different for everyone. It's really this understanding of which pathway is altered, which one is dominant or a non-redundant, where we will answer. So I think it will not be one of them, it will be all of these in different patients.
So thanks again, Stefan, for coming and presenting.